Since it's 1:30, welcome to all of you who joined in time. And we go ahead with our first speaker, Professor Damia Laoui, who graduated in bioengineering sciences and obtained a PhD from the university, the VUB, the Free University of Brussels, in 2014. After postdoc at the ISREC Institute in the École Polytechnique de Lausanne, she came back to Brussels and started leading her own team focusing on tumor immunology. He's exploring the role of immune cells in tumor progression and is developing new personalized combinational immunotherapies based on dendritic cells to win the battle against cancer. To share her passion of science, she is actively engaged in science communication, specifically encouraging young women to start and pursue studies and careers in STEM. Professor Damia, please, the floor is yours.
Okay, thank you for the nice introduction. You do see my slides, right?
Yes, I do.
Okay, so 90% of the cancer patients that die from cancer actually don't die from the first tumor they get. So they don't, they die from metastasis when the tumor goes from one place to other organs, or they die from tumor relapse. So when the tumor comes back after they were cured several years before. And the issue is that against metastasis and tumor relapse, we don't have a lot of therapies that work well. So we really need to understand how cancer works so that we can find better therapies to save actually 90% of the patients. And so how does cancer start or how does it work? Well, it actually starts with the cells of our own body. For example, here, like a cell from a breast. And this cell can then be transformed in a cancer cell. And there are a lot of ways how this transformation can occur. Usually what happens is that the DNA in a cell will get mutations. And these mutations can occur by different ways, or because of people that smoke too much, or because exposure of on the UV radiation by the sun, sometimes also by virus infections, or sometimes these mutations can also be inherited. So it can be that you carry mutations in your DNA that you got from your parents. And since our cells are also renewed in our body, it can happen that just by bad chance you get a mutation while your cells are duplicating. And so what is the difference between such a transformed cell and a normal cell? Well, a transformed cell, a cancer cell, will continuously multiply, even if it's not needed, but it will multiply until you get a small organ that will grow in your body, like a small tumor organ. And there are already some therapies that can kill cancer cells quite effectively, such as chemotherapy and radiotherapy. But these therapies, they will kill all cells that are multiplying. So they will kill cancer cells, but they will also kill other cells in your body that are multiplying. For example, even if you don't see it, now my hair is growing. And so it means that my hair cells, the keratinocytes, are also multiplying. And so if I get chemo, then it will also kill these cells and my hair will fall out. And so that's why people will also get quite sick from these types of chemotherapies. But so when you think about a tumor, so it's something that is growing, like, for example, a bad plant that is growing in your nice garden, there are two ways of tackling this. So, or you really attack the plant and the seed, or what you can also do, if maybe for this you need very toxic things, you can also maybe try to make sure that the soil around the plant is not fertile anymore. And so by attacking the soil, so what is around the plant, you can also destroy this plant. And that's a bit what we will start to do with our tumors. So we started to look at what is around the cancer cells. And then that was when it was realized in the field that when you look at a tumor, it's not just a full amount of cancer cells together. It's much more complex, and it looks a bit more like this. It looks like a small organ, which has obviously cancer cells inside, otherwise it wouldn't be a tumor. But it also has structural cells that keep the tumor as some kind of solid tumor. It also has blood vessels, and these blood vessels are interesting for the cancer cells because they bring oxygen, they bring nutrients, and so this will help the cancer cells to live, to survive. And it has a lot of these other cells that my lab likes a lot. These are the immune cells. And so what are these immune cells doing there? What is actually the goal of our immune system? Well, our immune system has three main functions. The first thing that our immune system has to do is to recognize everything that enters your body that is foreign and that shouldn't be there. For example, if your body encounters a bacteria or a virus that should not be there, this will be recognized by your immune system as something that is non-self. And so this is the first thing, recognizing what is foreign. The second thing is, the second goal of your immune system is once you have recognized something that shouldn't be there is to kill it. And the third function is to remember this thing. So that if the same bacteria or the same virus enters your body again, that you don't get sick, but that you immediately remember, okay, this was something that shouldn't be there. We will have already everything in place to kill it immediately without you getting sick. And that's a bit also the principle of vaccines, where you already put your immune system in contact with something foreign, a piece of something foreign, so it doesn't make you sick, so that your immune system is already trained to attack the real bacteria or virus when it comes in your body. So if we then have this tumor, we have different types of immune cells here. And so we wanted to know which cell is doing what. So who is good in recognizing, who is good in killing, who is good in memory. So we have to understand what are the roles of these different cell types. And the role of a cell is defined by the functions that a cell does, and functions of cells is defined by the proteins that are produced or expressed by these cells. So we have to know what are the proteins that specific cell types in the tumor express, and the proteins that a cell can make are encoded in the DNA of a cell. So the DNA, probably you all know what DNA is. It's a very long molecule that is in a cell, and this DNA can then be transcribed to what we call mRNA, so messenger RNA, and this will then be translated to the proteins. So if we know which mRNA, and so this is the message to make these proteins, is expressed by every cell, we will know, first of all, which cell it is, because mRNA also carries the identity of the cell, and we will also know which function these cells can do. And all these mRNAs is what we call genes. So we have to know what are the genes that are expressed by every one of these cells to understand what their function is. And if you look a bit to this tumor, you can see that this is a mixture of things. It looks a bit like a cocktail. And so if you have a cocktail and you want to change it, we want to also change the soil around the plant. So we want to change, modify the cocktail. We want to understand how it is. To change it, we need to know what are the ingredients. We don't only need to know which ingredients are there. We need to know how many of the ingredients are there and also what their function is. Like bringing something sweet or bringing alcohol or bringing something acid. So this is is the information that we need to understand, and this is what is encoded in the genes. So we take our tumor cocktail, and we don't need to know information on the cocktail, but on each ingredient. So we'll use techniques to separate all cells. And so we have, we work together with civil engineers that can make this type of techniques where you can split your cell one by one. And you see, this is an oil in water emulsion. You know, if you mix water and oil, you will have these little droplets form. So we can push cells every time in one such a little droplet. Okay, so we put our cells from the tumor in such little droplets, and we will also attach a colored bead so that we afterwards can recognize which cell, the components of the cell, which color they had. And so what we will try to do, I will not go too much in detail here, we will actually look at what is the DNA in each little droplet. And this DNA, we will then transform it to RNA, and then we will understand actually what are the genes that are encoded in each of these cells. And since there is a barcode and this color, we can retrieve from what cell these barcodes originated, for what cells the DNA originated. So what we then do is that we apply some bioinformatics, and with this bioinformatics, all these genes will be able to identify cell and to cluster the cells according to their functions. And what we will see is, first of all, which cells are there. So we'll be able to unravel the ingredients that are there, how many there are, and where they localize to understand also their function. And for example, here you can think this cocktail was probably bad because it had way too much lemon. And you see that these ingredients are sorted according to their functions. So everything which has vitamins is here on the bottom. Everything which is sweet is here at this side. Everything which has alcohol is here. Everything which is tasteless is here. So according to the identity, to the genes, you can cluster the cells in identities. And so every single dot that you see here is a cell that comes from a tumor. Here, this is an example of a lung tumor. So each dot is a cell, and then you can make these little clusters based on their RNA. And you can also then look at what genes are activated and what these cells are doing, because based on the genes that they express, we know which functions they express, and we then also know what these cells are doing. So if we go back to the cartoon, using this RNA, so the technique I just showed you is called single-cell RNA transcriptomics, we can give identities to the cell and also understand their function. And one of the cells is the dendritic cell, which is this cell here. And this cell is very well good in recognizing what is foreign. Remember, the first function of the immune system is recognizing what should not be here. The second cell that is here, it's a T cell. It's actually a soldier. It's very good in killing what should not be there. So this is the second function of the immune system. But so how do these cells now work together in a tumor? So dendritic cells, these are very cool cells. They are the whole time floating in your body and searching for what shouldn't be there, trying to recognize something foreign. And so if you have a tumor, then they will enter your tumor. And once they enter your tumor, they will capture, so they will recognize what is strange. So they recognize a cancer cell, because cancer cells are mutated, so they are recognized as being foreign. The dendritic cells will be able to capture a piece of this foreign material of the cancer cells to have some kind of a proof, so that they carry the proof. This is called a tumor antigen. And then they will bring this to their headquarters, which are the lymph nodes. And in the lymph nodes, they will present this piece of tumor antigen to the soldiers. Basically, what they want to do is to show the soldiers, look, I have a proof that there is an attacker, something foreign, something bad in our body, and you have to generate an army to kill this. So that is what is then happening in your lymph node. And so these T cells, so the soldiers, they will multiply until they form some kind of soldier T cell army. And this is something that you all have already felt in your lymph nodes at some point. For example, if you get a virus infection in your throat, you get a pharyngitis, you will also have your dendritic cells that will take parts of these viruses, bring it to your lymph nodes here in your neck. And here you will then feel your lymph nodes that swell. And this is just your soldier army that is formed. So this happens also in cancer. So then your T cells, your soldiers, will go back to the tumor. And there in the tumor, they will recognize the cancer cells and they will specifically kill all these cancer cells. So there are more cancer cells being killed, more material that comes available for dendritic cells. And this circle goes on and on and on until all the cancer cells are dead. So it can be that some of you who are now listening to me, and maybe Hilde and maybe other people, have a small tumor in their body now, and that we don't know of. And that's actually not an issue as long as you are healthy, because our immune system is doing this and is killing these cancer cells. And by tomorrow, these tumors will be gone. But so this is something that is constantly happening. But as we know, patients get cancer, so sometimes it can go wrong. And there are different ways where in this cycle issues can happen. One of the things is that these T cells, the soldiers, sometimes they don't function well, they don't work well. It can also be that sometimes they try to do their job, but they are suppressed by the environment. So there are other things close to them that hamper them in killing. Or it can also be that the dendritic cells and the generals of the immune system are not able to recognize well what is actually happening, what is foreign. And so that's where we will start to use immunotherapy, which basically only means boost our own immune system to fight against cancer. And to give you some examples on what we can do to tackle these issues, if you, for example, look at this thing like T cells don't function well, what we know is that T cells, when they get activated by the dendritic cells, they are ready to kill. But the fact that they can kill so easily cells in our body can also sometimes be a bit dangerous. So that's why our body has in the T cells also imprinted some brakes. And these brakes are put there so that in case these T cells go crazy and start to kill everything, we can also, let's say, pull the brake so that the T cell stop to get activated. The issue is that the cancer cell has also understood this, and the cancer cell will then put some proteins on their surface to pull the brakes of the T cells so that the T cells lose their weapons and start to die or stop killing them. So yeah, the cancer cells are just exploiting these breaks. These breaks is what we call checkpoints, so immune checkpoints. But luckily, some very important scientists that also got a Nobel Prize some years ago, Jim Allison and Tsuku Honjo, they understood this in the early 90s and they thought, okay, if we don't want this to happen, so then if we can block these interactions, for example, by using antibodies that block the interaction, then the T cell will not stop fighting, will continue to fight the cancer cells, and so then we are sure that they continue to function well against tumors. And so this is an immunotherapy. So these antibodies that block this interaction, which are called immune checkpoint blockade therapy, that is now already currently used quite a lot in the clinic. So if you know people that have immunotherapy against cancer, it's probably this type of therapy that you're getting now. And for example, for lung cancer, in a lot of cases, this is even given already in the first line, so before chemo or radio. So this is one example how we can use immunotherapy to boost this cycle. Another issue is that sometimes these T cells are functioning well, they want to kill, but they are suppressed. And that is because in the tumor, there are not only cells of our immune system that act well, there are also some cells of our immune system that are supposed to help us, supposed to help the body, but that they got corrupted by the tumor, such as, for example, these macrophages. And a macrophage inside the tumor, we call it a tumor-associated macrophage. And what was seen is that in a lot of cancer types, like all these here depicted in red, if you have a lot of macrophages in these cancer types, this correlates with a very bad prognosis. So we knew, okay, these macrophages are usually really bad guys. And the other issue is that there are plenty of macrophages in such a tumor. If you look again here when I showed you all these little cells in a tumor, this full blob of cells, so which is the major compartment of the cells in a tumor, are actually macrophages. So we really have to understand also what T cells are doing and why they're acting bad. And as you can see here already, there are several colors of macrophages, probably because maybe they can do maybe different things. So when we go back to biology and immunology, which types of macrophages were already known? Well, macrophages were typically classified in two flavors. So this is an oversimplification, but it's maybe easy to explain what they can do. The first one is called an M1 macrophage. For example, I have actually a cat that is sitting here. If my cat starts to, play with my screen, it might also start to scratch me. My cat came from outside, so it will put some bacteria in my skin. What will happen? I will get a kind of inflammation here. I will have a wound. and this bacteria will start to grow. This is not a nice thing. These M1 macrophages will arrive, they call M1 because they're first, to arrive in my wound. And what they will do is that they will very quickly start to kill all these bacteria that are there, and they will make an inflammation. We are calling them pro-inflammatory. And so I will have some swelling. It will get hot here on my wound. And this is very good to kill everything which enters your body. But it also provokes some tissue damage because they just kill everything. So after a while, all my bacteria are killed, but I have now inflammation, I have a wound. So what I need now is another wave of macrophages, the M2 macrophages. And what they will do is that they will start to heal my wound. So they're anti-inflammatory, they will stop the inflammation. All the pieces of dead cells, they will start to degrade them. And they will also make my blood vessels that might have been damaged nice again and remodel my skin and so on. But if you think about this in a tumor context, here you have a killing machine that kills everything. And here you have some kind of cell that tries to make the situation nice again. This means that if you have this in a tumor, it will start to kill the tumor. That's actually what we want. While this will help the tumor to heal and make nice vessels in the tumor and so on. So again, I said there is the M1, M2. In real immunology, you have plenty of flavors in between. But so we know that in our tumors, most of the macrophages are these M2 ones, the bad ones that start to heal the tissue. And what they do is that they suppress immune activation. They will suppress the soldiers, T cells. They will also make new blood vessels because that's what they are good at in trying to make everything nice. And making new blood vessels in a very quick way is something that we call angiogenesis. And this is also a bit of an issue because, first of all, your cancer cells will get even more nutrients, they will get more oxygen. But these blood vessels, they will be made very, very quickly. And if you do something very quickly, usually you don't do it well. So these blood vessels will be full of holes. And the cancer cells will kind of see this and will think, oh, a blood vessel with a hole, let's go inside, jump in it, and use this to go outside of the tumor to other places in the body. And that's a big issue because that's what we call metastasis. And that's when your cancer cells go from one place in the body to other places in the body. And that's where a lot of patients, can't be treated anymore. So we have to do something against these macrophages. And so several years ago in my lab, we started to study these and we saw that both of these macrophages, and so the good ones and the bad ones, they come from the same precursor. It's called a monocyte. And so if we want to get rid of these bad macrophages, there are two things that we can do. Or we try to kill these, or we try to make sure that this precursor only becomes of the good one. So these are strategies that we can look at. So we thought, okay, maybe let's try to see how we can potentially kill these bad ones. To do that, we have to understand how we can recognize them from the good ones. And we saw that they were overexpressing some markers, some proteins on their surface, such as this marker, which is called, the name doesn't matter, it's called macrophage mannose receptor. But so this is specifically expressed here on this surface of these markers. So what we did is that we made things that could bind here, that could specifically recognize these macrophages. And as maybe you have heard of nanobodies, so these is very small types of antibodies that were discovered in my lab before I was born. And so, yeah, in our lab, we use plenty of nanobodies. And so we made nanobodies that can specifically recognize this marker, MMR, that is on the surface. And then we can add something to the nanobodies, for example, a radiolabel that can, for example, kill the macrophages or also visualize it. And so when we inject mice that carry a tumor with macrophages, with this nanobody against macrophage mannose receptor, this nanobody will then specifically bind on these bad macrophages. And what is quite cool is that you can then also use this also just for imaging tumors or to image the bad content of macrophages in the tumor. And I will see if this video works. Maybe not because yeah. So this is a mouse that carry a tumor here. And so you can nicely image the tumor, and this tells you, oh, this mouse has a tumor with a lot of macrophages, so it's really a bad thing. And this is here a mouse, but so now since some years, we already have this also in the clinic. And so we use this for breast cancer patients to kind of show how many macrophages these breast cancer patients have. And the reason why it's very important to also be able to show this, we know that these macrophages can suppress the soldiers, the T cells. So we also then know that if you will just give immune checkpoint blockade therapy to the soldiers, so the thing that boosts the T cells, it won't work because they will anyway be suppressed by these macrophages. So this permits us to give the right therapy to patients and not just give any therapy and overtreat and lose a lot of time, which is very precious for a cancer patient, and also lose a lot of money. So there are ways that we found now to specifically kill these macrophages. Another thing that we can do is make sure that all monocytes that enter the tumor become the good ones. And so therefore, we try to understand what is actually dictating the fate of this precursor. Why is it becoming a good or a bad one? And we saw that is dependent on a specific receptor that induces signaling in this monocyte to become these good ones. And if you have a lot of, so basically, if you bind a receptor, we will block these macrophages to become the bad ones. So we could also generate some types of molecules that could also bind these receptors. And if you have this, you will make sure that all the monocytes become the good macrophages. So basically, we were able to have techniques that are, some of them already in the clinic, some of them are still in the lab, able to repolarize, as we call the bad macrophages, into good ones. So again, here, we know why T cells sometimes don't function well, but we can also find ways to using immunotherapy to overcome this. Then a last issue I talked to you about was the fact that maybe these dendritic cells, so the ones that are good in recognizing what is bad or what is foreign, maybe sometimes they don't work well. And so before I started working on dendritic cells, what was thought in the field was that anyway in tumors, dendritic cells are not able to recognize and capture these pieces of tumor cells, that they were also not able to move to the headquarters, to the lymph node, and that in the lymph nodes they were not able to give the information like to boost the generation of a T cell army. But I think when you want to be a scientist, you have to be very stubborn and not always trust what is already known and maybe see why maybe some things are not always correct. And so what we looked at was to understand, is there only one type of dendritic cell, or maybe there are different that do different roles? And that's also something that we found, is that there are different types of dendritic cells, CDC1s, CDC2s, also here one which is called monocyte-derived dendritic cell that come from the same precursor as a macrophage. It's maybe closely related to a macrophage. So we found different types. And then the next question, okay, you have different types of dendritic cells, but what do they do? So we wanted to see who is actually able to recognize cancer cell. And there we saw that, well, the three of them can still recognize properly what is foreign and can also capture tumor antigens. And then when we looked at who can migrate to the lymph nodes, where there were only these two that were able to do that. This MODIC was not able to do that. And if you then look at who can activate soldiers, who can activate T cells, we also saw here that only the CDC1 and CDC2 can do that, while the monocyte-derived dendritic cells, they can't do that. But on top of that, they were even good at suppressing these T cells, so at inhibiting immune reactions. And this was actually an important finding because people were already starting to use dendritic cells in clinical settings, but all these therapies were not working. That's why the field was thinking dendritic cells in tumors are bad. The reason is that they were all using this monocyte-derived dendritic cell because they are just more present in much bigger numbers than these other dendritic cell types. And so because the fact that people have been using this bad dendritic cell for years in clinical settings, dendritic cells were, got this type of name of not really working in tumors. Okay, but so we understood that these monocyte-derived dendritic cells were actually behaving a bit like corrupt macrophages and were also able to suppress these soldiers. But the good thing is that we could also find that there were two types of dendritic cells that were still doing well, that were having anti-tumoral properties. And we also wanted to know in which types of tumors do you have these good dendritic cells, and we looked in different mouse models, and we saw, and so the black here and the gray here are the good ones. In actually all tumor types that we analyzed, we had these nice dendritic cells. Also, when we looked at different types of patients' samples, so different types of tumors in patients, again, here we found that the good dendritic cells are present everywhere. So in all types of tumors, you have good dendritic cells. And that's very nice because this means that maybe we can use these dendritic cells for therapeutic interventions. And so that's what we did in the lab. So as a first experiment, we want to see, can we use these dendritic cells to vaccinate mice against getting tumors? So this is a control group. So we inject tumor cells in a mouse. What happens? Well, the mouse will have a tumor. But what would happen now if you first vaccinate a mouse with dendritic cells that come from a tumor that already carry this antigen and that can already stimulate an army? We first tried this with the CDC once, and then we saw, oh, if you then inject a tumor, it's very cool because these mice are protected and tumors are not growing. And we saw that this worked nicely for melanoma. And we also looked in these mice, there were much more soldiers that were generated. Then we thought, okay, let's try this also with the other type of dendritic cell that was behaving well, the CDC2. And we saw again that in some tumor models, these were also being able to protect mice from tumor regrowth. So again, here, if you pre-treat with dendritic cells coming from tumors, if you then inject tumors, the tumors will not grow. And we saw that this, for example, worked nicely in lung cancer. And we saw that this was hampering bad macrophages to be generated. And what is our dream or hope is to also do this very soon in patient setting. And so the idea is the following. If you have a patient with, for example, here a lung tumor, this can be surgically removed by the MD and by the doctor. And from this tumor, we can then isolate the right dendritic cell that has carried all the information and that has captured the material from the tumor cells of this patient. And then we can re-inject this dendritic cell in the patient that now doesn't have a tumor. And what will the dendritic cell do? It will do two things. The first thing is that it will move towards all places in the body, and because remember, these dendritic cells, they like to swim in your body, and they will go to actually try to find whether there are also what we call micrometastases, so very small metastases that you can't detect with the current technologies like CT scan or MRI. So they will be able to already kill small metastases, but also they can generate a memory response, so they can generate T cells that can have a memory response so that within, let's say, 5 or 10 years, the patients will not relapse and will remain healthy. And so we hope to be able to use this tumor-derived dendritic cell as a kind of personalized therapy against metastasis and tumor relapse. So we now have data in mice. We don't know whether it will work in patients, but we are very hopeful. Obviously, to do this in patients, this costs a lot of money. And I don't know if you know this cartoon. So I asked Santa for a research grant. Do you still believe in research grants? Well, I don't know if I still believe in research grants, but I do believe in Santa because I did encountered a Santa who is called Yamina Crossa. So she is an ex-cancer patient who started a fund. And so she's now also raising money so that we can also do this clinical translation. And so I started doing my research surrounded by these very nice and very um, known and very very good researchers. But I always thought if I would at some point have my own team, I would like my team to look a little bit more like this. And so this is my team today, and I really thank them for being there. They are now in the lab while I'm here giving a presentation. And so, yeah, these are all fantastic people, and I really thank them a lot for all the work they do. And I thank you for your attention. And yeah, if you have any questions, I'm happy to reply to all questions you might have.
It's Thank you so much, Professor Louis. It's amazing how you can translate complex science into something that is so easy to understand. I'm of course a biomedical scientist, so maybe I'm privileged, but I think um, also the economists and those who are not familiar with our immune system really could understand what you are doing, how you really work towards a targeted therapy. We do have a question. I hope, Ghana, that you're here. I saw your question appear. Yes.
Yes, hello. Maybe I was too fast with asking my question because I was interested in how would you address trans differentiation that is happening between macrophages and one and two. But I guess this is not your field, as I understood later. So if you're focusing on dendritic cells, I guess my question would be how it would be plausible with the concurrent ongoing therapies for cancer patients. Because, for example, if they're taking chemotherapy or something else, you would have elimination of dendritic cells. And how would you address that?
Mhm. Yeah, it's a good question. So actually, the good thing is that dendritic cells are quite resistant to chemo and radiotherapy because they don't proliferate once they are mature. And since they do not proliferate, they don't expose their DNA. And so we have seen that if you have a tumor where a lot of cancer cells are being killed, dendritic cells are very resistant. What we are doing now, so if you will do a first clinical trial, we won't be able to do the trial with patients that don't get the standard of care. So we have to combine in a safety trial the dendritic cell therapy with standard of care. So we are now assessing what are the effects of standard of care or the dendritic cell function. And we see that, for example, chemotherapy has not a lot of impact on the dendritic cell function. What it does, it can, because it kills cancer cells sometimes by something which is called immunogenic cell death, it will release more antigens, and so it can even more activate the dendritic cell in the right way. So we will anyway have to do it together with chemo in the first trials, but our hope is that if it works really well, that potentially in a later stage we could give dendritic cell therapy and stop having these patients getting chemo. That's a bit what the hope or the aim is. And to reply on your first question, so what was it? How we repolarize macrophages?
No, but I guess you're not focusing on macrophages, but there is such a thing as a repolarization of macrophages, as in one turn into two. For example, when there's a resolution of inflammation, M1 becomes a M2 and vice versa.
Yeah,
So just by killing M2, I mean, if there's M1, they might become M2 eventually.
yeah. So the thing is, if they would become an M2, so macrophages are long-lived cells. So they're very plastic, and so they will really respond to the environmental stimuli, and they will be able to repolarize. If you would fully kill your tumor, and so you don't have an inflammation anymore, then you don't mind to have M2s. Because if the cancer cells are gone, then you would actually be happy that your M1s that did the job repolarize to an M2 to also heal the remaining ex-cancer tissue. But typically, that's not what is happening. Macrophages on its own, even if you repolarize them fully, it's not enough to revert, as a cancer therapy. So a lot of cancer therapies in clinical trial with molecules that do repolarize macrophages, but these seem to not be effective as such. You always have to combine it with something else. And we don't fully understand yet why they don't work as effectively as we see it happening in mice, for example.
Um, but you're using humanized mice, right?
No, we use immunocompetent mouse models.
as an immunocompromised, right? Like
No, immunocompetent. So we use mouse cell lines in fully immunocompetent mice, or we use patients. But we
but
don't.
wouldn't it be I mean, mice immune system is different from human.
Yeah, but that's also the big issue, eh? So what are the differences? It's not that different. For example, dendritic cells are extremely well conserved in between species. For macrophages, is a bit less. But that's also one of the reasons why in the last years, the 2, 3 last years, a lot of funding stopped going to oncology because a lot of mouse models were giving extremely nice results. But then when they were going in a phase 1 in patients, it was fine. They were not toxic. But in a phase 2, they were not showing the expected results. And that's why there was a big shift in funding, and a lot of companies shut down their oncoimmunology field. Now it's going back again. So now we see that all the funding that went to infectious diseases is going a bit small by small back to the oncology field. But there are differences, but we don't always know exactly why. And a lot of effort is also being generated in making better cancer models, where the tumors are not only tumors that you inject, subcutaneously, where there's like one mutation in the tumors, but where you have spontaneous models, spontaneous tumors generated in the mouse that really recapitulate the different phases that you have also in patients.
Back in the days, I worked in cancer research about 20 years ago in the States, and we had rat models that were basically they didn't have immune system to begin with. So that's how you could grow a tumor in a rat and then test the
Yeah,
drug.
I mean, xenograft models are being used in mice, but then you can't, assess the immune system, huh? Because
Right.
you typically don't have a functional T cell compartment. So,
That's definitely a challenge.
yeah,
Thank you.
you're welcome.
Thank you so much, Damia, for your presentation, for your answer. You said that you were very close to the patient setting. When can we expect?
So it depends a bit, let's say, on the money, because scientifically and ethically we are ready to start. So now we are writing a lot of grants. And so we hope that if we have the money, we start early 27. And if not, it would be early 28.
Okay. I hope Santa's there soon.
Yeah, thank
I
you.
know you need to run. Thank you so much for your nice presentation, and thank you for the time you spent with our students.
Okay, thank you. Yeah,
Thank you so
have a
much.
nice day.
Bye.
Bye-bye.
You too. Bye. And then I immediately will introduce to you Professor Liesbeth Geris, who's a professor in biomechanics and computational tissue engineering at the University of Liège and KU Leuven. Her research focuses on the multi-scale and multi-physics modeling of biological processes, together with her team and their clinical and industrial collaborators. He uses these models to investigate the etiology of non-healing fractures, to design in silico potential cell-based treatment strategies, and to optimize manufacturing processes of these tissue engineering constructs. She's currently executive director of the Virtual Physiological Human Institute, and in that capacity, she advocates the use of in silico modeling in healthcare through liaising with the clinical community, the European Commission and Parliament, regulatory agencies such as EMA and FDA, and various stakeholders. Thank you so much, Lisbeth, for being with us, and please don't hesitate to start presenting your wonderful research.
Thank you very much, Hilde, for this extensive introduction. Can you see my slides and can you hear me well?
Perfect.
Yeah, okay. Oh, I just lost them myself. There we are. Good. So I guess this will be quite a change of pace because what I will be talking about is mostly not the research that we are doing ourselves in skeletal tissue engineering and bringing that to the clinic, but it's the work that I've had the honor to lead. It's a community-based work that we've been working on since quite some time in order to make sure that whoever is developing in silico-based tools and technologies, that the whole pathway is ready to roll this out and bring this to the clinics and the patient if they're ready. So that's basically what I will be talking about today. And rather than talking about in silico medicine, the new buzzword in the field is digital twins. So the use of digital twins to support patient-specific optimization of clinical interventions. And maybe since this is quite a diverse group of people, I will first spend some time on explaining what is a digital twin and what are some examples of digital twins that are in the clinic or very close to the clinic already. And then I will I will give some background on the broader perspective. So what are all of the aspects that we need to make sure are in place in order for this new technology to reach its full potential for the patient and for society. So let me start with an explanation on digital twins. And I don't know if people here have heard about the concept of digital twins, but what digital twins is the use of computer modeling and simulation in health and care. So very frequently people equate this to AI or things like that. And if you then ask what are the ingredients for something like this, then they will say data. So that always reminds me of a brownie, as you see here on the slide, where the brownie would be the insight that you gain from a model or AI, or the advice that you give to a physician, or the input that you give to a patient. And then, of course, the data that you are collecting about the patient are the ingredients. But very frequently, discussions then turn into something like this. If we just have enough data, so if we can make all of the electronic health records available, if we can collect all the data in a database, then magically, somehow, this brownie will appear. Now, of course, that is not the case. What you need is a recipe in order to make the brownie. And there can be many different versions of recipes in the same way as there can be many ways of making sure that you take your data and you transform it into actionable knowledge for a specific stakeholder. Now, digital twins are such recipes that you can use. Digital twin is not a new concept. It actually comes from the engineering field, where it is used quite a lot already, and where there is a very strict definition of what a digital twin is, namely that it is a virtual representation of a physical object or a system that follows it throughout its lifetime. And what is very important is that this physical and digital twin are in constant communication, so that there is a sensor readout from the physical twin that feeds the digital twin. And then there is also the digital twin then does calculations and then gives feedback to the physical twin and is even able to automatically or semi-automatically recalibrate this system or this product. Now, if you think about this in a context of health and care, this is very difficult, right? In how many cases would you be able to real-time measure all of the data that you need in order to make a good prediction about a certain process? If we think about cancer, like Damia talked about, or the immune system, like how would you read out the relevant information from that based on sensors? It's not possible. How would you then, even if that would be possible, how would you dynamically recalibrate your cancer? That's also not something that happens. So when we started applying the concept of digital twins and silico medicine in healthcare, we came to this broader definition that says that it's the use of individual specific models for all aspects of the health and care process. So from prevention, prediction, diagnosis to treatment follow-up. And not just that for a patient, but also the evaluation, optimization, de-risking, selection, and personalization of intervention options. So drugs, devices, advanced therapies. Now, what is important for this concept of digital twin is that there is this link between the physical and the virtual. But in health and care, this will be through human-in-the-loop decision-making. So people will be making measurements on patients or on organs. This will be interpreted, and this will be translated into data that goes into the virtual twin. And then the virtual counterpart will make a prediction that is then interpreted by somebody as a clinical decision support system, for instance, to make a decision on the physical part. Why do we insist on using this concept of digital twin? It's because it is, when we talk to patients, it is very powerful because it gives a clear message that this is something that has information about you and can be used to make predictions and to do tests on something that represents part of you digitally, so that the experimenting can be done not on yourself, but on your digital twin. So that makes it an interesting concept to use. What is important to know about digital twins is that, of course, there are different components, and there is always a hardware component, so sensing, imaging, anything that will allow us to collect data. And then, of course, also compute, because we need to be able to calculate these prediction models. So we need high performance computing or edge computing or bedside computing. There is then the software on the other end that has the model of the twin, that does the simulations, the analysis, and then there is some middleware that makes this all come together. What I will be focusing mostly today is the in silico part. So in silico refers to silicon, the main component of computer chips. So when we talk about in silico, or the term in silico was a bit established as a counterpart to in vitro and in vivo in the lab or in living beings. Good. When we talk about digital twins, this do not need to be digital twins of you entirely. This can be a digital twin of a cell, of a tissue, of an organ system, or of the entire system. It can also be digital twins of therapies, and I will explain this movie a bit further down. It can be digital twins of manufacturing setups, where we come a bit closer to the original definition in the engineering industry. And what you see here is actually work from our lab, where we created a digital twin of how cells grow, that can capture how cells grow in a 3D environment in a perfusion bioreactor, where we can calibrate it with sensor readout, but then use the twin to get a better idea of what the environment is that the cells are feeling and the extracellular matrix that they're producing that you see here in pink. And then there are digital twins of medical facilities that allow to do to optimize the logistics supplies, but also during COVID, the stream of patients, so that there was minimal crossover between groups. Also important to notice is that for digital twins, we do not make an a priori on what kind of digital technology will be used to create a twin. So if we put all of the in silico technologies on a scale, and you have on the left-hand side the black box, it's a very old term, but the AI-based, the machine learning, the data-driven technologies, where we don't make any assumption on things that we already know. If you look at how this is applied in health and care, or where this has the most power, it's when you have large sets of data that have a certain quality that is there. So in image analysis, huge contributions have been made by AI to assist radiology in decision making. But if you think about other situations, then frequently we have lots of data, but it's very heterogeneous and it's also not well annotated. So then it becomes very difficult for AI to do something with that. On the other hand of the spectrum, we have what we used to call white box models, where we start from knowledge, physics, physiology, chemistry, biology, mechanisms and laws that we know translated into mathematics. Now, this is also not very frequent, because in biology, mechanisms are known until they're not known. So we know something at some level, but there's lots of things around it. But in physics, it's more easy. And actually, everybody is already using, in clinics, is already using white box models, because if you go from the raw output of your microscope or of your CT MRI device to the image that you're actually seeing, this is already a processing that involves physics, that involves mathematics, and so that uses a model. So everybody's already using models. But most of the models that we look at for digital twins are sitting somewhere in between, where there is a part of the things that you know, part that you derive from data, and in where you use AI technologies on in silico data, where you use in silico to have constraint on your AI. So there's all kinds of combinations. The point is that when we look at creating digital twins, we must select the technology that is most appropriate for the question at hand, the knowledge we have about that question, and the data we have that is relevant to that question. So to make this a bit more concrete, what are digital twins? And I repeat that these examples are examples that are very close or into the clinic. The first example is from colleagues from the University of Amsterdam, where they are combining patient-generated data from their smart devices with electronic health records and with other medical information that is available from the patient. This whole data set is then compared to with an existing broader data set of other patients for which they have also the longitudinal data, of course, that have gone into cardiac issues. So for which there were cardiovascular issues that prompted urgent care. And by making that comparison, what they aim to do is detect, even before the first disease onsets are there, detect risks. And that way inform the patient and go towards a active care rather than a reactive care and a very early preventive care. Now, what is also important about this initiative is that typically patients that are gathering this data, they fall in a certain socioeconomic category, let's say. So it is important, and that is what colleagues in Amsterdam are also doing with their solutions, is to make sure that this gets tested and rolled out across the entire population. So they have selected the different neighborhoods or looked at the different neighborhoods in Amsterdam, making sure that people are there from every cultural, socioeconomic group or background to make sure that this app becomes as powerful as possible. A second example comes actually pretty close to what is the engineering definition of a digital twin, and that is the use that is glycemic control in the intensive care unit. Now, patients in the intensive care unit, they don't eat, so they are on a certain nutritional scheme, and then the nurses have to check regularly whether they have too high or too low glycemic levels in the blood, and then correct either by changing the nutrition or by correcting with insulin. Now, this, of course, takes time, and the fact that they do this at intervals makes that the glycemic levels of the blood actually fluctuate in these patients. Whereas we know that hyperglycemia delays the healing, we also know that hypoglycemia, so not enough glucose in the blood, is even worse. So they try to get a balance, but ideally we would have a constant glycemic level in the blood. Now, what this tool does is it took information that we have on how glucose gets transformed into metabolites that we know from basic chemistry. They translated this in equations, and then they used a part that is data-driven because every patient in every kind of situation responds differently to insulin that is provided. And that doesn't really, there were no equations that could really capture this. So this is done data-driven based on the response that they had in the past on insulin provision. So this hybrid model, let's say, then allows, and you can test it, there is a spin-off from the University of Liège called InSiliCare. If you go to their website, there's actually a tester where you can play around with the levels of nutrition and insulin and see how this influences the glycemic levels in the blood. So this is now in a clinical study in the University Hospital in Liège, where the nurses use the readout from the patient. The model gets this, the model makes a prediction, and they can actually test for the right level of insulin to give to the patient to have the longest or to avoid fluctuations. In time, ideally, this would become something that becomes semi-autonomous or autonomous to reduce the burden or to reduce the workload on the healthcare providers. At the moment, what they see is that with a more optimal glycemic control, patients are able to leave the ICU a day early, and that is long-term stay patients, which is already a big cost saving in intensive care. Another example is that of surgery planning. In this case, it talks about the left atrium appendage occluder device. So that is this LAAOD. What does it mean? Well, we all have an appendage, or we can have an appendage to our left atrium. It has different shapes. And what happens in patients with atrial fibrillation is that this is the location where blood clots are formed. These blood clots can then travel to the brain, and 1 in every 3, roughly, ischemic strokes are actually caused by blood clots originating from this place. So what they didn't do is either give anticoagulants as a therapy, or they simply block this appendage so that nothing can get in or out of this appendage. Now, since there are different shapes for every patient, there are also different occluder devices, different sizes, and they can be placed under different angles. So what surgeons do is they look at the medical images ahead of the surgery, make an assessment on the size and the attack angle, and then go in. Now, 1 in every 5 cases, this was not the right decision that was made pre-surgery. And so they have to withdraw the device and put in another device, which means that they are using 2 devices for 1 patient where only 1 is reimbursed. So this is an extra cost. What people are now doing, and these are images from a spin-off of Barcelona University, but there is also a spin-off from the University of Ghent, FEOPS, that is now acquired by Materialise, that has this already approved for clinical use and reimbursed in some countries in the world. They make a model of the left atrium where they can simulate, here you see the appendage, they can simulate the occluder device, the size, the positioning, and then they can evaluate here how much blood is still able to enter under those conditions and adjust. And they've seen this, so colleagues of FEOPS have done the clinical study, and they've seen that they go from 1 out of every 5 that requires a second device during surgery to 3 in 100. So this is a serious reduction and almost complete removal of these second devices used. Another example is pharmacogenomics. There are over 600 drugs on a list at FDA that have a genomic biomarker, which means that if you have a certain sequence in your genome, that would mean that you can maybe be a fast metabolizer or slow metabolizer, or that you could even have an adverse response to certain drugs. And if you look, for instance, in Germany, there are some numbers that indicate that I think 6.1% of all the admissions in hospital are due to adverse drugs responses, which costs 4.5 billion euro a year to the healthcare system. So if we can already, based on people's genome, prevent lots of these adverse events, well, that would be very beneficial. But not just that, also people with different responses might have, rather than going through the different meds that are available, immediately choose the right one based on your genomic digital twin. Another example where the digital twins are used not directly on the patient or by the healthcare provider, but by companies creating healthcare products is this one. This is a lung digital twin. The lung part is based on the patient geometry, but then you can have the alveoli, that is, those are more idealized geometries. And how this is used, for instance, is in the optimization of the design of these inhalers. If you look at the lung, so basically an inhaler injects all kinds of particles. They travel then down to the lungs, and they need to reach every part of the lung. Now, if you color code all the parts of the lungs and then work back using the model, what is the trajectory, or where are these particles coming from? For instance, the ones that end up in the yellow zone, where are they coming from? Well, it turns out in that specific design of inhaler, they're coming from this upper right quadrant, which means that if you're using your inhaler slightly wrong, then there is a part of this that ends up in your mouth rather than in your lungs. And so part of the lungs will never, ever be reached by any of the drugs. So that is not what you want. You want that however the patient uses the device, so however little of the drug is actually reaching the lungs, that when it reaches the lungs, it reaches all parts of the lungs. So you want all the colors to be mixed in this cross-section here of the device. So what they did is they played with the design, so the length of the inhaler bit, and then also the speed with which the particles are released in order to get designs that have a much better mixing of the color, which means that even if only a tiny fraction of the drug reaches the lungs, it will reach every part of the lung. And then there are examples of clinical studies where these tools are used, for instance, to augment clinical trials when you have rare diseases, of which we have quite a lot, over 6,000 rare diseases. So this is a universal immune system simulator where we have an agent-based model. So every cell is an agent. They have a specific characteristics, which means that they're governed by a specific behavior and they can interact with other agents that they encounter. This simulates the immune system. In this case, this is applied to tuberculosis. So there's also a part of the human lung and the response to the tuberculosis, to the tuberculosis disease maker. Now, what this then does is, in this case, this is used to optimize the design of a clinical study that looks at vaccine development or testing of the vaccine. These people have tested so they've trained their model, they've shown that they're able to capture relevant biomarkers of the disease and of the response to the vaccine. And they went to the European Medicine Agency to get qualification advice for their immune simulator. They've gotten a letter, they've got to the stage of letter of recommendations, which means that they're well on their way, but they need to answer a few more questions before they get qualification advice. And the intent here is to really use this between phase 1 and phase 2 clinical trial, to prepare the phase 2 clinical trial to have a better design of the trial that is optimized for the patient population. And then the last example where we have an example that's already from 2018, where an in silico clinical trial completely replaced the human clinical trial. It's a very specific case though. This is a pacemaker. We've all seen when you go to MRI that there is the sign, no pacemakers allowed, which is why, because here we have electricity going through this lead wire. And when you put electricity or a wire that leads electricity in a magnet, you get electromagnetic fields that are formed around it, which will heat up the tissue from the inside. Now, what this particular company did is they used their knowledge of physics, their previous models of how the pacemaker functions, to design a lead wire that was kind of shielding the electricity better so that the variations of the electromagnetic field around the lead wire would never be more than the variations that we have between humans. And they tested this design in a salt bath. So first in an in vitro setting, then they also did one animal test that was already scheduled for something else. And then they simulated a virtual cohort of patients, different sizes, and also different locations of the pacemaker, because it has to go behind the collarbone, so it curves quite a bit, and that also influences the physics. And in total, then they also simulated different scanning sequences. So 2.3 million different scenarios in total were tested in silico, and for none of that, the predicted variations of the electromagnetic field were beyond what you normally see in patients. They put this to the regulator, FDA in this case. FDA reviewed this. They were already familiar with the model. And they were convinced by the credibility of the analysis that were done, because of course also this is physics that we're simulating. We're not simulating biology with more uncertainty. This is physics, laws of physics that we've known for quite some time. And they allowed the company to go to the market without doing a 250 patient 2-year follow-up clinical trial, which meant, of course, that they could save quite a bit of money to not have to do the trial, but also that a lot of patients were able to get the device sooner, which then also was making more money for the company. Now, again, this was an example of a first case where there was a full replacement of the human clinical study. There haven't been so many examples of that since, because we're talking here about physics and not about the biology that they were modeling. Good. Now, if you look at all of these examples that I just gave you, they're focused on one specific organ apart from the immune simulator, with one specific scale, length scale. So these are actually still relatively simple digital twins where we can where showing the credibility is more easy. Now, we have many more complex diseases. We have comorbidities that we want to address, and people are already working on digital tools for that and on silico models for that, but there's a lot of reinventing the wheel. So, and also a lot of that reinventing the wheel is funded by the European Commission, where when a project stops, sometimes the knowledge or the tools simply disappear. So they wanted to make sure that this didn't happen, and that is why they created a new flagship that was inaugurated at the end of 23, called the European Virtual Human Twins Initiative, where they want to accelerate personalized care through the use of digital technology in line with the European values. And so they want to have a collaborative system. They want to have they want basically a place where people can collaborate, where they can share their knowledge, and where you can exchange resources, data sets, models, algorithms, workflows, etc., connecting also to the European HPC infrastructure, and doing this in a way that complies with the EU rules and values. And that's so that is basically what what I was involved with or got involved with a couple of years ago. That was through the I'll skip this just to be a bit faster. The EDIT project. EDIT is a coordination and support action that is over now, where the European Commission asks us to bring together the entire ecosystem related to digital twins in health and care and write the roadmap for the realization of the virtual human twin. And so we started doing that. We talked to a lot of people from all kinds of backgrounds about their opinion, their needs, their expectations, their fears, their concerns related to their own healthcare journey and their expectations on digital twins. So we started with patients, civil society organizations, healthcare providers, as they are the beneficiaries and the users of VHT technologies. So VHT is virtual human twin. We talked to the creators, research innovation actors, industry, often also represented by their communities. We then talked to infrastructure actors, the funding financial actors as the enablers of those technologies, and then also the managers, so the governance, the ethical, legal, social issues. So that was quite a diverse group of people because there's not one patient, there's not one industry actor. They're very diverse. And in total, over 800 people contributed in one way or the other to the roadmap that we drafted. Yeah, I mentioned the use of ecosystem organizations or societies. They typically really are able to translate the experiences of the users and the technologies better to the policymakers and vice versa to have that communication flowing. And one of them is the one that I'm involved with, the Virtual Physiological Human Society, which represents the public and nonprofit actors. So what is this in this roadmap? And we're now in this roadmap looking at, we followed, in order to set this up, we followed the same strategy that we would also follow for any medical device. So we started from the need basis, talking to all these people, asking what their need is. This, of course, is in the context of the current trends, the healthcare cost, the imperative, so personalized medicine, the push of AI, the digital agenda of Europe, sustainable development goals, including health, but not limited to health. We talked to industry and what they would want, so small, big companies, developers, CEOs, software, medical devices, drugs. And it really came down to these 7 points. So they want to have access to resources. They want to use these kind of infrastructures created by the European Commission as sandboxes or benchmarks, follow up on new technology development, get more clarity on ethical, legal, social issues, especially the legal issues, and then networking. Whereas when talking to clinicians, they focus mostly on things that make their work and their professional life and their work-life balance better. So addressing unmet needs, improving patient outcomes, reducing the cost, enhancing the care of quality, with the big remark that, of course, this needs to be done in a way that is integrated in the clinical workflow seamlessly, access needs to be very easy, and data annotation is always a challenge. So this needs to also be facilitated. And maybe I'm going a bit fast now, but in the pre-reading material that I sent, there is the roadmap where you can read each of these different parts, the ones that are of interest to you, and you can skip some of the others. Normally, it should be very readable that way. We also looked at the technology and the infrastructure that could realize something like this. So the technology meaning what is already available, what models are available, what infrastructures are available, and what is still necessary. And what is still necessary is a sort of advanced simulation platform where you can log into like you would on any platform. You can find their repositories of models or data sets, or through this interface, you would find catalogs of other initiatives that are already collecting models or data sets. You would be able then to put together a workflow, so run a simulation, having access to the HPC centers, so the high performance compute centers across Europe, and be able to run validation workflows. And that will allow you to build up the evidence that you would need in order to go to the regulator later on. This should be done, of course, in collaboration with already existing initiative, the European Health Data Space, you heard about it, and other initiatives that on a more scientific level or basic level are collecting images, are collecting data, and are collecting research results. So how would this then work? We look at this as you would have a data space that for every data point you have information on where this is coming from. Is this a clinical study? What were the patient groups? What is the information? And then the models would be data space crawlers. They would crawl across this data space looking for data sets that actually match their requirements so that they could run and make a prediction for these groups of people. And this predicted data would then also become part of the data space. Then we worked quite a lot on the ethical, legal, and social issues, starting with, for instance, standards. The challenge in this case is that we have so many different medical problems that we need a wide variety of digital twins, of in silico solutions, using different technologies. So having one standard is typically not possible. So we first listed all of the standards, and there are already over 150 standards available that have to do with something related to digital twins, with the data, how we collect it, how we store it. Some standards make reference to models, but not so many. So we thought maybe we should make a new standard for digital twins only. And I'll give you a second to read this. So, of course, we cannot make a standard to rule them all. We are making a new standard, though, that focuses on credibility assessment, because that's still very important when going to the regulators. How should they assess credibility? And they are lacking at the moment a standard that does that. There is an American standard for medical devices that puts forward principles of verification, validation, and certain quantification that are actually valid across the globe, but also across different medicinal solutions. So these principles are now taken forward, and we're putting this in an ISO IEC harmonized standard. We're also talking to notified bodies, to the European Medicine Agency, to make them understand how to do credibility assessment of these technologies. For that, we also worked on other practices. You have this translation from government, intergovernment, non-governmental organizations that all create requirements that get translated into good manufacturing practices, good laboratory clinical practices, and then gets down in the organizational QMS or SOP. But there's no such thing as a good simulation practice. So we thought, let's put something together. Now that there's more attention, we need to inform people of how to do good simulation practice. So we started writing a white paper, but so many people joined from all kinds of different backgrounds, as you can see here on the list, that in the end, we ended up with something that was 80,000 words long. So we published this as a book that is freely available, and it's actually a good first introduction into how to do credibility assessment, what are the aspects that you need to think about, and where can you find further information. We then also did a lot of work with legal scholars to better understand what are the challenges currently in the legislation, the MDR, the AI Act, the European Health Data Space, how they are interacting or not, because there is often a clash between the AI Act and the MDR. There is uncertainty, especially for these digital tools. And also propose some solutions. And there we were actually quite lucky that while we were doing all this work and we were talking to policymakers and the European Commission, that they also decided to rewrite the MDR. So in the previous or in the currently existing MDR, there's only one mention of in silico medicine, which restricts it to preclinical use. That is often used to not look at the in silico evidence. But now we were able to convince them that this is something that needs more space and needs more words in the regulation. And you can see it in the proposal for the revised MDR that they actually included much more evidence or reference to in silico tools, including not as clinical data, but as part of the clinical package. So for this, we had to do some lobbying. This is very different if you're a mechanical engineer to go and lobby politicians, but it's an interesting exercise. And I'm almost done now. We focus a lot on the ethics also. Well, we started working on the ethics. We didn't go as in depth as we did on the legal. There are, of course, the ethical legal challenges and the autonomy, the questions on equity. Is the use of digital technologies going to make the equity and the equality in health and care worse or better? There are arguments for both, but we need to be attentive for this. And then some of the broader challenges when you put your philosopher hat on, looking at digital twins, is this being or having? So there are some interesting questions to ponder over in the evening. Then we did do a lot of work on the social side, especially on trustworthiness and trust, because if you look at the difference, what as a developer you can do is look at validation, credibility, explainability, transparency, but you need to really also work on creating a certain level of awareness, because without understanding, you cannot have trust. Without understanding, it is blind faith, and that is not what we want. So we did develop a lot of educational material. Also with patient organization, we developed the Code and Cure series, where if you still don't know what a digital twin is or what an in silico model is after this presentation, please go have a look there. It's in very short, simple movies explaining the different components of a model and how it could affect the health and care journey. And we put all of these learnings into an info kit so that if you want to do focus group with a patient or a Delphi survey, that you will find everything from the source material to the emails that you need to send, the ethical approvals that you need to apply for in this info kit. We worked with colleagues that are focusing on other technologies like VR to do planning, teaching, communication, to show ways of how this can then be translated to the patient. There is capacity building, education that is required. So some modules were developed to explain in silico to the basic, to people in training still, so master level, but also retraining. And then we worked on the uptake. So we looked at the market, and these numbers are whatever they're worth, showing that the interest in digital twins is rising. But we need to be aware that we need to always have the need there, that it's not just a push from companies that are seeing this as a way to monetize the healthcare system later on. No, there needs to be a clear link to the need. So we need to ask the right questions. And then we worked on incentives. So what does success look like? Are we able to reduce the cost? Probably not, but maybe at another, not in the healthcare, let's say, portfolio, but by making people return to work-life faster, we might be saving on other aspects. So quite a bit of things that we did there. And then we grouped this into 30 recommendations across these different aspects that I just mentioned. Now the project is over. You can read all the conclusions in the full roadmap. We had a policy brief that is slightly smaller because, of course, nobody's going to read 300 pages. There's also a 2-page version, which is called the manifesto, that you can find online, that we had signed by over 130 organizations, and not me, Lies from KU Leuven, but it was actually the vice rector, the CEOs, CTOs of companies that are signing this. And it's a good way to remind the European Commission that they started this thing of the virtual human twin, so they better keep supporting it. And then we're still engaging, continuously engaging, for instance, in the health policy platform of the European Union, with further stakeholders like the payers and the buyers, so healthcare technology assessment or health technology assessment agencies and insurance companies, to really look at the adoption of digital twins. And we are now building this advanced platform. We are part of the consortium that's called VHTNet, that is at the moment now, we just kicked off the user requirement elicitation sessions that again will talk to all of these diverse backgrounds so that we make sure that what is developed is there for everyone. Because that's the whole point of this technology or of the work that I presented, is that we want to rise the tide, because that will lift all of our boats, whether it's a research boat or an industry boat or a patient boat. The goal is to make everybody's life better. So thank you very much.
Thank you so much, Liz, for the presentation. I'm very, very grateful to be in touch now with you, because I didn't really know what to expect, but I knew I got the recommendation from somebody who presented last year, who did a fantastic job. So I trusted that this would be good. And my God, you exceeded my expectations. I immediately understand, well, not only what a digital twin is, of course, but how important your work is. You very clearly showcased how important this collaboration is with different stakeholders, because on your own, nobody can bring technology to the patient on its own. So I'm very, very happy that you showed indeed patient engagement, community, citizen participation, that this is super, super important. I don't see the questions, but if there are people here in the panel who have questions, don't hesitate. You touched upon a lot of things that we already discussed. The regulatory sandboxing indeed this morning, as well as an incentive. And indeed, I think you rightfully said it, that there needs to be a need. Otherwise, we don't speak about innovation. Then it's just pushing an invention to the market where nobody's waiting for. So I'm utterly, utterly grateful for this presentation. I learned a lot. And I so understand why you got 2 ERCs. It's amazing, your work, and thank you for all what you do to push health technology reach the patients. Thank you so much.
You're very welcome. It's an honor to be able to talk about this, of the work of so many people. And I find it super exciting, like you say, to work with so many diverse profiles. So it's yeah, I'm very grateful. I'm happy also to take any questions later on. I can imagine that I talk too fast and too much. So
no, not at all. But this year the cohort of program participants is a little bit more quiet, I would say. But if we get questions, I will definitely transfer them to you. Thank you so much for your time spent with us.
Very good. Thank you so much.
Thank you, Lisa. And then I will immediately introduce Isabelle Francois, who holds a PhD in bioengineering and possesses extensive experience in innovation, strategy, and IP management within the life sciences and healthcare sectors. As an IP officer at KU Leuven Research and Development, she managed a significant patent portfolio and facilitated spin-off creation through strategic advice. She was a director of innovation and strategy at Medvia, and she recently co-founded Givana to provide strategic guidance in digital society and healthcare innovation. And she serves as managing director at Health House, driving growth and partnerships. He has various advisory roles, including chair of the External Advisory Board for the Digital Society Institute at KU Leuven, and she co-chairs the Healthcare Committee at XR Valley. Academic credentials include this PhD in bioengineering and a degree in bioengineering from KU Leuven. Isabelle, I'm very, very honored that you're here. I introduced what you will speak about very briefly this morning, so I won't take any longer from your presentation. Please, you can.
Yeah. All right. Hi, everybody. Very happy to be here with you on this Friday afternoon. So yeah, very happy to be here. Listening to you, Hilde, just reminds me of the fact that I'm not getting any younger, but yeah, it is what it is. So yeah, maybe to add a bit to the introduction, I'm a bioengineer in cell and gene biotechnology at KU Leuven. Quite a while ago, did a PhD in applied biological sciences. I have a kind of academic background, that's for sure. But today, I'm not going to talk about any science or any scientific progress or any scientific inventions, innovations. So I heard the last two speakers and thought like, oh my God, I will be the odd one out here this afternoon. But so please hear me out.
No. And Isabel, maybe, sorry to interrupt, but I really, and this is so important for the students as well, to understand that to facilitate innovation, to reach the patient, it needs a whole ecosystem, including supply chain and manufacturing, and making sure this delivery from inventions gets to become an innovation. And it's really the whole process. So don't feel, please, the odd one out. You are needed with your projects too.
Well, very happy that you say so. And to be honest, I realized that when you have this incredible innovation, this incredible treatment, that's the first, that's of utmost importance, but you need to get that treatment to the patient. And for me, it was given. You know, you go to the hospital, for instance, or you go to your pharmacist, you just ask for the specific treatment you need, you get it, you take the medicine, and off you go, and you get better, hopefully. But it was in 2024, I attended a conference on advanced therapies. And advanced therapies at that time were defined as cell and gene therapies and tissue engineering. And I will come back to that later on. And there was a big panel discussion. And the conference actually was focused on these therapies, but also on the logistics part. There was a big panel discussion with all types of different people there, including a hematologist of KU Leuven, Koenraad de Bakkere. And he was sitting there on the stage, listening to people coming from the logistics, coming from supply chain. And he actually said, and that really stuck with me, well, I'm the clinician administering these cell therapies, it was a CAR T therapy, to the patient. And to be honest, I never thought that getting the treatment to the patient, so getting it to me as a clinician, is so hard, because he heard all these logistics players talking about the struggles they have to get a package from A to B to C and vice versa. So he said, I never thought about the struggles that or how hard it is to get that therapy to me so I could administer it to a patient. And that's when it struck me like, he's right. They're absolutely right. I didn't think about it either. So I was very happily surprised that last year I got involved with specifically a logistics project at Brussels Airport. And that's what I'm going to talk with you about. And I prepared some slides, not that many, because I'm more of a talker, I have to say. So let me share you the presentation so we can start talking about this. I'm assuming that you're seeing my screen.
It's perfect.
Okay. I'm not using Zoom that often anymore, so that's why I was asking. So the project, as any project needs, has a title which is called the PTLG project, Precision Therapy Logistics Gateway Project. Now that's a big, it's a big mouthful. And let's first look at the precision therapy part. What are we meaning with that? Because logistics, you understand, it's about logistics of a specific therapy. But with precision therapies, we mean this cell therapy, gene therapy, tissue engineering, but also radioligand therapy. And I'm going to, because this is a very diverse group of people, right? So I'm not really sure what the amount of expertise it is that you have with the different therapies. So I prepared some very simple slides to explain these different therapies. And the first one is gene therapy. And for people from Belgium that are in the audience, they will, if I speak about baby Pia, they most probably will understand what I'm talking about. So baby Pia, it's the little baby that you see here, 6 years ago, she got into the news. And why? Unfortunately, she got into news because she has a rare genetic condition, which is called SMA1, or which is called by defect in gene SMA1, and which leads to spinal muscular atrophy type 1. Unfortunately, that's a genetic disorder. It's a rare disease that cannot be treated very easily by normal like medicines, by an oral drug. No, it needs to be treated by gene therapy. So it's a gene in the body of this little baby that was affected, and that could only be restored by, well, actually, or be cured by actually adding the correct gene, and that correct gene will replace the damaged gene. And well, there you go. That's a gene therapy, replacing a defect gene by then the correct gene, the correct mature gene. What's the big issue, you think? There is a cure. What is this all about? Well, the thing is, at that time, that treatment, one single shot with that gene, with the vector carrying the correct gene, that treatment, one treatment, it costed about 1 million euros. And it was not reimbursed by the RISIF, our agency. So you can imagine, as a parent of that little baby, you're looking for all kinds of way to get funding. So they got in the news because they needed crowdfunding to be able to really administer that therapy to their baby. Long story short, they got the money. So the crowdfunding activity, it really worked. They got the money, and now 6 years later, baby Pia is a toddler. She is, well, healthy is maybe a big stretch. She has some developmental problems, but she's alive, where normally the life expectancy of little babies like that is about 2 years. So she's alive, she can go to school, she has some developmental disorders, like she doesn't walk that easily, but she's still there. So that's one of the therapies. So you hear already, life-saving, often for rare disorders. It's very valuable, but also very costly. So a very valuable, costly therapy, which you can imagine it's very important to get to the patient in time. Now, the funny thing is, I'm a freelancer, so a consultant. I do different types of projects. And this morning, believe it or not, I talked with the clinician at the University of Antwerp that actually is treating baby Pia, that really discovered that baby Pia was suffering from this rare genetic disorder, and that treated this the little baby. And when I was talking about this afternoon, about speaking with all of you, and saying like I was going to explain gene therapy and well, also the logistics part of it, she said like, well, oh my God, that's so what a coincidence, because I treated baby Pia, but also because she said like, we are dealing with these rare disorders where gene therapy often can be the solution, but we often realize, or we often encounter problems with the logistics of these therapies, meaning that the transport is not done in a timely manner. The transport is not taking account the really very precise temperature restrictions that are needed for that therapy, so that often when the therapy then finally arrives, it's not useful. It cannot be used anymore. So you see, there she really emphasized the need for a correct logistics. So just as a side note. Second example, or second part of the precision therapy, is what is cell therapy. And I show here an example of a specific therapy, which is called CAR-T cell therapy. And this CAR-T cell therapy, maybe it rings a bell for some of you. It's treated, or it's used for treatment of certain blood cancers, also certain other rare types of diseases. And now it's under investigation to treat autoimmune diseases, like, for instance, lupus, which is a really very peculiar skin disease. So what happens? If you have a specific type of blood cancer, and I have to say, these cell therapies are all often used as a last resort, meaning that you need to undergo first other therapies like chemotherapy, for instance. But if you have been treated with all these other therapies and it doesn't work, then often as a last resort, this CAR T cell therapy is being used for specific blood cancers. So what is being, or how does this therapy work is, well, you see the picture here of the patient. Well, you see the patient here. It starts all with apheresis, which is just meaning, or it entails that T cells, which are a part of, or a specific T cell population, actually, which is a specific population of immune cells in our body, they are being collected from the patient. And these T cells are then being transported. So this arrow actually means transportation to a production facility, which is called here Make CAR T cells in the lab. It's transported to a production facility of a pharmaceutical company, like, for instance, J&J. They have a very big production facility in Ghent, which is called the Legend production facility. So the T cells of the patient, the very sick patients, don't forget that, often used as a last resort. Patients are very sick, so it needs to happen fast. So these T cells are being transported to the specific production facility of a pharmaceutical company somewhere in the world. They are transformed in these CAR T cells, which you can see here. So it means that they are genetically modified with a gene that will ensure that a specific receptor is being expressed on the surface of the gene. And that specific receptor, CAR, the chimeric antigenic receptor, so the CAR receptor, that will recognize specific antigens on the surface of the cancer, of the blood cancer cells. So this T cell is being genetically modified to produce this CAR T cell. These CAR T cells are then multiplied again in the lab of that production facility to have millions of CAR T cells. That again is transported to the clinic, where the CAR T cell is then given as the last resort treatment to the patient. So it's often used as last resort, I already said it, but also a one-time treatment, meaning a one-time infusion of the patient with these CAR T cells. These CAR T cells then start multiplying also in the body of the patient itself, and then hopefully are attacking the blood cancer cells in such a way that these blood cancer cells get eliminated. And even years after the first initial transfusion within the patient, you can find, even after years, you can find these CAR T cells in the body of the patient. So very valuable, last resort. You hope, of course, if you're the parent or a relative, or if you know somebody with this type of blood cancer, you of course hope that this cell therapy works, because last resort, also the patient needs to undergo, before getting this CAR T cell therapy, a very, very strict treatment with immunodepressants. So making sure that the other T cells of the patient are being removed, so that these CAR T, these engineered T cells or have the ability or have the possibility to really grow in the patient. So very important. And you see here, we're talking about live cells. So meaning that we're talking about the transport of living material that needs to be transported very quickly and under very strict temperature controls. So important to remember that we're talking about very expensive, valuable therapies under very strict conditions that are being or have to be used by the transportation firms. Another possibility, or another part of these precision therapies, is tissue engineering. And here I show you the example of Holoclar, which is a tissue engineered product. And we're looking at people that have a specific eye condition where the outer parts of the cornea, which is called the limbus, are not functioning very well, and they lack a specific stem cell, which is located there in the limbus, that stem cell is not functioning correctly and is present in very low amounts. So what is being done here is that there is a biopsy being taken, so stem cells are being taken from the healthy limbus, also here shipped to a production facility where of that corneal part, cells are extracted, being put into culture, meaning they are being cultured so that they can again multiply to a very large number of these cells. And it's being done with a primary cell culture, secondary cell culture. It's being then shipped after they reach a specific amount of these tissue engineered cells. It's being shipped then to the clinic again, where under surgery, the correct or the new stem cells are being placed back into the cornea of the patient. So you see here again biopsy of cells, so living cells being extracted, being multiplied, and then being shipped back. So again, we're dealing with living material. That means you cannot leave it out in the open or out at high temperatures for a long time, because then the cells die, and you realize, of course, that means that a patient cannot be treated. So very important. Also here, as with the CAR T cell therapy, you see it, we're talking about cells coming from a patient and being manipulated in what kind of way, and then being transferred back into the patient. So it's not only time is of the essence, not only temperature is of the essence, but also the chain of identification, because you don't want to have cells from another patient being brought into your body. And that's a very important element. And it seems very easy, but it's not. For logistics, it can be a nightmare to make sure that chain of identification throughout this whole process, and also throughout the CAR T process, is being maintained. So a very big challenge there. The last specific type of therapy, which we are calling, we see as part of precision therapy, is radioligand therapy. And it involves radioactivity. And for instance, maybe your father or somebody else in your family or your friends have had prostate cancer. I for sure hope not. But for prostate cancer, for instance, there are different types of treatments, and radiation is also one treatment. Radiation coming from the external, from external sources. Of course, when you think about radiation, you think about putting these specific type of radioactivity or specific type of radiation on the surface of a person. And that means you can delineate a tumor, or you can try to delineate that very specifically. But if you have to radiate from the outside, you will always damage also healthy cells surrounding the tumor, because you never, ever can really radiate in a very specific, localized manner. So you will always affect healthy cells outside of the tumor. And you can say, well, it's a price to pay to also affect then the healthy tissue. Yeah, that's one way of looking at it. But there's also an alternative. And that alternative is, for instance, radioligand therapy. Not for all tumors, of course, but for specific tumors, such as prostate cancer tumors. And of course, if your prostate cancer is metastasized throughout the body, and I will show you an example later on, you cannot radiate all these types of different tumors within your body after the tumor has been spread out in the body. So then that radiation from outside is not an option, and you need to think about really creating a kind of nuclear missile, you could call it. I don't like to use that word actually, and especially now in these times, but you could look at it like that. It's really a kind of molecule that will look for the tumor inside your body, and that's that molecule that is looking for the tumor inside your body is what we call the cancer targeting ligand. So it's a molecule that will recognize specific proteins on the outer sides of the cancer cells. It will link with it, and then that specific molecule, as you can see here, is linked with a radioactive particle, and you have different radio emitting isotopes. And I will, in the next slide, talk about actinium-225, which is an isotope that emits radiation very close by. So really 1 or 2 cell layers is being reached or being targeted by that specific radioactive particle. So you can, by the combination of this cancer targeting ligand with this radioactive particle, you are really targeting only the cancer cells. You bring the radioactivity very close, in very close proximity of the cancer cells. So as you can see here, the radioligand together with the radioactive particle is internalized by the cancer cell, where the radiation is being released. The DNA of the cancer cell is being damaged. The cancer cell will die, and as such, these specific types of cancers can be removed from the body. And for me, this example is really mind-blowing, and it's an example already from 2016. So it's not that very new, I admit, but the thing is that this has been used in a clinical trial, and the fact that it's not being used commercially currently is because of that there was the need of looking for a way to produce actinium-225 on a larger scale, and that is being found now. And we can be as Belgians, we can be very proud of the fact that this really big amount of actinium-225 that is needed for commercial use is currently being produced locally in Belgium by Pantera, which is actually a company situated at the SCK Sense site in Mol, of all places. So their actinium-225, thanks to the work being done there, can be used more commercially in the near future. But that being said, for me, the example that was mind-blowing is the patients that you're seeing here. It's a male patient who suffered from actually prostate cancer, but as you can see, all these black dots is the metastasized cancer that originated from the prostate cancer. So having PSMA, which is a specific antigen on the surface of the cells, not only within the prostate, but also here throughout the body. And then for me, really mind-blowing, after 3 times injecting that specific therapy, so the cancer ligand, which is a ligand recognizing PSMA, together linked with the 225 actinium, after only 3 times administrating this specific therapy, you can see that this cancer is completely removed and the patient is completely cured. Then there was a follow-up treatment. You see after 7 months, the M is months, not minutes. And after 10 months, another kind of control treatment has been, or supporting treatment has been done to make sure that the patient remains cancer-free. So really mind-blowing therapy. So I don't think I need to convince you all that these precision therapies, as mentioned, the gene therapy, cell therapy, tissue engineering, and radioligand therapy, that these are life-saving treatments, often used as a last resort. They're very valuable. They also cost an enormous amount of money because often, like the CAR T therapy, it's one patient specific, and the transport is extreme time and temperature critical, and with this very important chain of identification that is needed. And within Brussels Airport, and I'm representing Brussels Airport, we're currently looking at this PTLG project, meaning that we're looking at these precision therapies as the next or the future when it comes to pharma. And maybe you know that Brussels Airport is one of the most important pharma hubs in Europe. It's transported during the COVID pandemic, 1 billion of the vaccines were transported via Brussels Airport, making it a very important airport. And for instance, I can give you an example. The Winter Olympics in Peking, they could not have been happening without the shipment of these vaccines via Brussels Airport, just to give you a kind of context there. And Brussels Airport, being currently one of the most important pharma hubs in Europe and wanting to maintain that position, that's wanting to move the needle forward, is thinking about future therapies and is thinking about these precision therapies and really wanting to evaluate whether it's important to create a specific infrastructure. Infrastructure, I mean a specific building. So bricks and stones, but also talents, hands and brains within the building to really make sure that the transport of these life-saving treatments is optimized. And the specific project where I'm project coordinator of is developed or divided in 4 phases. And the first phase was a desk research, an explorative study on the logistic of these precision therapies, like what are the parameters, what are the criteria, time, temperature, chain of identification, what is needed for the specific types of therapies, because a cell therapy needs to be transported under other conditions than gene therapy, for instance. And so there's a white paper out there that you can take a look at if you want, really looking into the logistics of these precision therapies. So that's one thing. Second phase, and that's what we're currently doing, is we're performing test shipments. And maybe it's important for you to know that in logistics, there are different types of players, and I will show them here. What I'm showing here is an overview of one transport actually going from A to B, and it needs to come back. Let me explain you more in detail. So we start at the University Hospital of Antwerp. With these test shipments, we're not using patient material, so it's not a CAR T cell therapy that we're transporting because it's a test shipments. We actually want things to go wrong to see where we can improve the whole logistic chain, but we don't want to do that with actual patient or treatment material. So we start in the University Hospital of Antwerp with cells of healthy donors. So really blood samples, serum samples, different kinds of samples that we, in the end, want to transport from Antwerp to biolabs within Dallas, so the United States. And here I want to show you, it's something I learned now through this project, because as said, I'm a bioengineer in cell and gene technology, so not a logistics expert, so I learned a lot, is that one important element within the logistics chains is what we call these forwarders, the freight forwarders. You can look at them as architects. So if you're building a house or whatever, you work together with an architect. The architect, he or she draws the plans, but also sees like, okay, which are the different elements I need to have this house being built. And that's what the forwarder does. So he says like, okay, you need to transport from A to B, from Antwerp to Dallas. Well, and you're in the hospital in Antwerp. Well, we need a truck or a van that goes to the University Hospital of Antwerp and that picks up the material of the patient. Not only that, we also need another transport, and it's not really mentioned very clearly here, but it's a packaging, because the university hospitals often, or the hospitals that are administering the treatment to the patient, often don't have these specific boxes that are being used to transport. So the freight forwarder, so the architect of the transport, gives a signal or does an order with this kind of packaging company. The packages, so the cardboard boxes or the styrofoam boxes, are being transported to the hospital via a truck or a van, something smaller than that. So they organize the fact that this is being transported. Then the van or the truck is going to the hospital to pick up the package with the samples and transport it by road to the airport. In the airport, you just do not drive immediately to the airplane. It doesn't happen like that. You have to give your package over. You have to deliver it to what is called a ground handler. And a ground handler, I will show it also later, is actually the owner of a warehouse where you come in with the package, you hand over the package to the people in the warehouse. They look at the documents, they see whether or not there are explosives inside and so on. So a whole kind of security check. After it's being security checked, then the ground handler is transporting via tarmac the package to the airplane. The airplane goes from Brussels to, for instance, Dallas, lands there, and then the whole thing again happens, but then in the other way. So the forwarder is the one engaging with the trucking company, is the one engaging with the ground handling company, and is the one engaging with the airport and the airplanes. So with Lufthansa or United Airlines or Qatar, whatever kind of airline you want to fly with. Important to know, this is not, but it is cargo material, but it's not being shipped by DHL, for instance. And DHL, as you might know, they have their specific planes, the yellow birds, the yellow planes. Well, we're not using the DHL, for instance, or we're not using these specific cargo planes. We're using passenger planes, and in the belly of the airplane, you have these packages of cargo being transported. So maybe next time that you're sitting on the airplane waiting for the plane to lift off, go ahead, look around, and you will see a whole lot of activity of transport or packages being put in the belly of the plane that you're sitting in. It's not only your luggage, it's also like pharma products being transported via that passenger plane. And this is actually just another way of depicting what I said. So what are we doing? So we are doing these test shipments with healthy or blood samples from healthy persons from Antwerp to Dallas and vice versa. There are companies out there freight forwarders that are really specifically dealing with these type of packages. And then you can say, like, why are you looking into this whole value chain or this whole logistics chain? Is actually these specific white glove forwarders, white glove companies, as we call them. They are very, and I mean very, very expensive. One package being sent, the transportation of it may cost around 5,000 euro to go from A to B, and then you have to come back. So very expensive. And we wanted to see whether you could use the other shippers that are being used now to ship, for instance, flowers. The flowers from the Netherlands are not from the Netherlands, they're coming from Africa, just as a side note, but to ship flowers or whatever, so that are more dealing with these kind of bulk shipments, whether or not we could use them in this process, because that would make the transport cheaper, meaning that smaller pharma companies, smaller biotech companies can also more easily use this kind of transportation means. Just to give you an overview, these are all the partners involved in these specific test shipments. So you see a lot of people involved in the project. Here, I just wanted to show you a small movie to show you the scale or the size of what we're talking about. So here, for instance, this is the University Hospital of Antwerp. You see the samples coming from the healthy donor, blood samples, or it's being stored in different -80s, which are freezers that are at -80 degrees. Then you see the little vial where then the other vial is being put into. We're talking here about cells, right? So these very valuable cells is being put in dry ice. We also include sensors, temperature sensors, and also geolocalization sensors to know at each point in time where the package is and what the temperature is, because we really want to stress test this whole logistics chain to see whether or not it's being transported at the right time in the right temperature. So to make sure, I can already mention or say that we have seen a lot of deviations already. So which means that this project is really very necessary. And the third phase of this project is actually looking into specifically that. So I mentioned that we have seen quite some deviations. So what we want to do is develop a standardized protocol, an SOP, and an internationally recognized, and sorry about the typo, transport label, to make sure that shippers, which are the pharma companies and the clinicians, that if they use a transportation or a forwarder or transportation firm that is being recognized, that has this specific transport label, we are sure that the correct time, temperature, chain of identification means or requirements are being met. So that's what we're trying to do. This is very unique. In the whole wide world, there is no such thing as this internationally recognized label. There is no SOP. So everybody is trying their utmost best to ensure that these transportations of these very valuable packages are being done correctly. But I can assure you there are a lot of problems. And lastly, and that's also something I'm responsible for Brussels Airport Company, we are looking into the fact whether there's the need of a specific infrastructure, a specific building to deal with these packages. And allow me to show you, and I'm first going to show you this. This is a warehouse. This is a typical warehouse that you can find at Brussels Airport, where you can see a lot of shipments. So you see a lot of these bulk shipments that are being handled within this ground by these ground handling agents within these warehouses. Now remember the movie I showed you, it's one box with one small vial. Can you imagine that box being put in here and then you need to hope for the best? I've been in these warehouses where I thought, how on God's green earth are they going to find my very small package within this bulk of the packages? So gut feeling wise, you can imagine that the creation of an infrastructure, a building only dedicated to these very small boxes is needed. Next to that, I also want to show you a screenshot of Brussels Airport. And if you go on a holiday, you are here, with different terminals. These are the planes at the terminal where you you board the airplane, and then you drive off to one of these, the strips, as they're called, for takeoff, and then you leave. We are talking about the cargo area of Brussels Airport, which is situated here. So here are the passenger planes, as you can see. This is the cargo area. I was talking about DHL. Well, they have their specific planes coming over here, where they are filled with cargo. So no passengers here, only cargo. If you zoom in a bit more, because you can even see, I hope that these are yellow. So these are really DHL airplanes. The warehouses I was talking about are situated here. So big warehouses where you can see that a van with a specific parcel, for instance, is coming here. This is Swissport, just for you to know. So Swissport is one of the big ground handling agents. So a van is coming here. It loads off the package into the warehouse. This goes through the warehouse, goes through all kinds of security checks and so on to make sure that there are no explosives on board, that the paperwork is okay, and so on. If that's all okay, they are leaving here, going via this road through an additional security checkpoint. Then they go here in a tunnel underneath all the strips and so on. And then they go to the place where the passenger airplanes are, and they are loading the cargo onto the passenger airplanes. And maybe it was a bit too much into detail, but for me, as a logistic novice, it was very interesting to see this process. So what do we want to do in addition? I was talking about this standard operating procedure. I was talking about the label. We want to take this a step further. So we want to actually look at different airports within the United States, also within Latin America, within the Middle East, within East itself, so within Asia itself. And we want to educate these airports, where we want to select a few airports, and we want to educate them to, learn them how to deal with these kind of packages, how to deal with this kind of transport, so that we know that, for instance, if we send a package from Brussels to somewhere, Abu Dhabi, Dubai, wherever in the Middle East, but also with a trained airport, that we know that we're not the only ones treating the packages as they should be treated, but wherever they land, that specific airport will also use the same SOPs, will use the same label, will use the same requirement and specifics as we do here in Brussels. Because what's the use if we are dealing with them very meticulously and very carefully to send them over somewhere else where they're left at tarmac in 40, 50, 60 degrees Celsius for the temperature to go up and your cells to die and the treatment is gone. And maybe the life of the patient is gone. So yeah, in a nutshell, these are all the partners I already showed you, but these are the partners that are also financially involved, also important to share. So that was in a nutshell what I wanted to talk with you about. And there you are. I was looking for you all. So yeah, that's no science, as you could see, but how are you going to transport such a valuable package, such a valuable treatment from A to B with in a correct way? So that was what I wanted to talk to you about. So thank you for listening to me.
Thank you, Isabel. It was very, very, and indeed, I think you turned us from logistic novices also in a little bit more knowledgeable. We do have a question, Julie. I'm not sure whether you're there, but I see your question.
I will look at the question.
You can also see it, yeah.
Yeah, but I see Julie there. Yeah, go ahead.
Yes, hello, good afternoon.
Hi.
Yes, I was wondering if I looked at the logistical pathway of these therapies, and I think they are all in small batches because you cannot really pool them because it's for patients in critical conditions. How much would it cost? Because it seems like it would be very expensive to do all this for just one sample at a time.
Yeah. So currently, the costs. So I asked the same question, right? Like, how much does it cost if you use now such a white glove service to go from A to B? Well, 5,000 euros per shipment. It's a lot. Big pharma companies, they can pay for it. That's not a problem. But we're looking also at smaller biotech companies with a lot of new novel technologies. Sometimes they're being acquired by a big pharma company, but say it's a smaller company, they have a problem to pay for that. And you could say you could charge a patient, but also patients have problems with it. So what we want to do is try to lower the costs. Also, you cannot pool indeed, but you could use some of these logistics providers that are not the white gloves services, but that can be trained to use the correct measures to then at a lower price, fingers crossed, transport the treatments. Sorry. I also saw your other question, right, on the production facility.
Yeah.
What we see is that, for instance, J&J, to give one example, they have a production facility in Europe, and they also have a production facility in the United States. So to ensure that the production facility is closer by to the patient. So there, also the price is being reduced because you do still need the different parts, but the flight and the time is less. So also there, that's a reduction of time. Galapagos, which you might know, is currently stopping the whole cell therapy. They had this decentralized production. They used a smaller production facility to put them inside the clinics, and for them, the clinics to create the treatment themselves. Well, apparently that business model is not viable because they needed to stop. I do agree
it
that
is because of the decentralized production, because I was thinking a lot of companies started investing in CAR Ts, but are now pulling back because it's not viable. So with our NGO, we're like currently looking at maybe promoting a social profit kind of production type to produce ATMPs or bring them to the market. But I was thinking, is this kind of transport like a sustainable way to do if you have like non-profit organizations trying to set this up, or?
I see what you mean. I think for a non-profit organization, it will be very hard, right?
Yeah, but if you like, if you look at collaborations between university hospitals and like external clean rooms like USA and Anicels, for example, I think it's only possible if you have some kind of decentralized production because they are small production sites, right?
Yeah, and there are indeed hospitals that are under the hospital exemption regulation are producing their own cell therapy. But of course, if you use a commercially available cell therapy, and you can only use hospital exemption if there is not a commercially available treatment present. If you use that commercially available, then you're stuck with the production facility of that pharmaceutical company. So yeah, it's not easy. It's not easy. I fully agree, and I hope that by turning the logistics pathway of those therapies, by turning them into something more or less costly, that can help. That could help them to speed up the process. Yeah, and we talked about CAR T, which is an ex vivo cell therapy. Now also more and more pharmaceutical companies are looking into the in vivo part, right? Where the transformation of the CAR T cell is not being done outside of the patient body, but inside the patient body. That makes the logistics part also quite, easier because you still have the temperature constraint vectors, but yeah. So there are hopefully some improvements coming along. Yeah.
Yeah. Okay.
Thank you, Julie, for your question. And indeed, I think this morning it was indeed raised that this hospital exemption, and it's particularly in Spain that they are exploiting this. Yes, indeed, to decrease the cost of CAR T cell therapy, but I think more broadly for ATMPs. And there is a study, but it's an older study because I showed it in the courses to the biomedical scientists, where the price, I think it was about Kymria or Yescarta, one of the two, was put on the market by Novartis, if I'm not mistaken, at 400,000 euro, and Doctors of the World, together with Health Policy Watch, they really calculated that you could make it for 60,000 euro. Of course, that brings us back to, they invested so much money in bringing this therapy to the market. Then again, we can link it to what we discussed this morning, this therapy, this transparency on what is really included in R&D costs for pharmaceutical companies. And I showed the news that for 7.8 billion euro, our Arcellx was bought by Gilead, and it's about CAR T cell therapy. Well, they put this 7.8 billion in their R&D cost, but that's mergers and acquisitions. That's not really R&D. So it's really, and I really, really like these discussions. It's all so linked, and there are so many challenges. And really, honestly, I'm so happy that you were here, Isabel, to explain that logistics. And I try to showcase this with the COVID example, that it's not only the science that can pose challenges, by far not. That really this whole supply chain and the delivery was in the COVID vaccines also a real issue because the mRNA vaccines didn't get to Africa because just don't have the means to make sure that the temperatures are being respected. So it's super important for, well, all the program participants to understand how complex this ecosystem is, and that really we cannot do it each working in our own silos, that we need interdisciplinarity. And indeed, well, you, Isabel, with your background in bioengineering and you understand how valuable and how needed this is, that this technology is respected, and that indeed temperatures and slight differences can really mean the difference between life and death for a patient. And although it's maybe one patient, but one patient is worth saving, and how valuable, and then indeed what the cost is for a patient's life. And it's, thank you for really, it's I'm very, very happy to have this this example to show how we can increase equity, health equity, really throughout the European Union, but more well, ideally, as I discussed this morning with students, globally, of course, that we can work towards a European health union, sorry, a universal health. So thank you so much. We went a little bit over time, but I don't think we ended an hour earlier than last year. So I don't think the students are upset about it. But maybe just to end, I want to thank you, Isabel. And I also wanted to say to the students that I know I promised to give you the division of the case study and the dates that you would be able to, or you needed to defend this case study, but we're still working on it because we have much more groups than we anticipate. We have 13 groups. We'll put them as soon as possible on the Université Virtuelle, as well as all the presentations that we got from the speakers. They will be as soon as possible, as soon as we receive them, we put them on the student platform. And I think if there are no questions left, I think we can conclude this day. Thank you for the presentation, really, for engaging us in this fascinating world. And thank you to the students who stayed with us until this very last moment. Thank you. And next week, I'm happy to meet you on site again.
Have a nice day. Bye-bye.
You too. Merci, Isabel.
Bye.
Bye.